Hi, let's continue our journey into the central dogma of molecular biology. So, just to recap, we said that information goes from DNA to RNA to protein, and these steps are mediated by two nanomachines. RNA polymerase II read DNA and write RNA, while ribosome read RNA and write protein. So today we talk about anemia, and let's dig into this concept. You have your gene for hemoglobin, okay? Into your sequence, your DNA contains a sequence of nucleotides that will result into the hemoglobin protein. So your genes is a sequence of nucleotides. So let's imagine adenine, cytosine, guanine, so on and so forth. At one point you will have these three nucleotides: cytosine, thymine, adenine. So why are they important? We will see later, okay? For now, just bear with me. RNA polymerase will read this sequence and will write RNA sequence. So when we have a cytosine, we will have a guanine, adenine, uracil, okay? For this triplet. There are nucleotides before and after. So our RNA will be exported from the nucleus into the cytoplasm, where ribosome can read it and make the protein. So this is the hemoglobin protein. I'm lying just a bit to you, because this is not a single protein, okay? For the sake of discussion, let's make it a single protein. In reality, it is a complex of 4 protein, almost identical protein, okay? So just trust me. Let's make it simple. So these triplets will be translated by the ribosome into this amino acid. Let's see, GAU. We go to the genetic code and we see that GAU code for aspartic acid, ASP. So in this position in your protein you will have this amino acid. So this is the normal wild type, it's called human hemoglobin. It's the hemoglobin that healthy people have. So now let's look at what happens when we look at people that have sickle cell anemia. So this is another chunk of DNA. Is another time this is the human hemoglobin gene, but now it's different. So let's look. Look, CTA. Now is CAA. So this is the gene for hemoglobin, but there is a mutation. So our thymine is become adenine. It's magic. It's a mutation, okay, into the sequence of the nucleotides. The RNA polymerase II, it has no idea of what a mutation is. It just read the DNA and write the RNA. So instead of GAU, it will read CAA and will write GUU. So our ribosome will read GUU, and instead of aspartic acid, GUU, our ribosome will read GUU and will put valine. So this is the form of the hemoglobin that people with sickle cell anemia have into their red blood cells. Okay, so what's the problem with this valine and this form of hemoglobin? The problem is that this form of hemoglobin is different from the normal, the healthy wild type form of the human hemoglobin. So this, when the concentration of the oxygen is low in the blood, will tend to form aggregate with other form of this protein, because this is like this amino acid is slightly hydrophobic. Okay, it's not important, but the thing is that this form of the human hemoglobin, when the pressure of oxygen, when the concentration of oxygen into the blood is low, tend to aggregate with other protein, and when you form a protein aggregate, the protein doesn't carry its function. So this form of hemoglobin often can't carry oxygen. So people with sickle cell anemia sometimes can have some problem on the carrying of oxygen. So, but you are smart. You are smart, and you know that you have 2 chromosomes. Okay, we are just seeing one chunk of DNA, but you in your cell have 2 copies of each chromosome. So, for the definition of 2 copies, you have 2 genes for human hemoglobin. So let's look at what happens. These are the product of your 2 genes in your 2 chromosomes. So this is the 3 different solution that you can have. You can have 2 genes for human hemoglobin wild type, so you will form two kind of human hemoglobin that are perfectly functional, and this is the situation where healthy people stand. Then you can have the healthy wild type form of human hemoglobin in one gene, in one chromosome, and the mutated form in the other chromosome, and this is the case where you are heterozygote. It's called heterozygosis because this is different. Zygote is like you got a different chromosome when you were conceived. So you have 2 forms of the protein, and these are the people that really one chromosome can stand. Okay, you are not so healthy, but you don't feel it. You can stay. I mean, well, okay, not perfect, but well. And then you can have all the 2 chromosomes with the mutant form of hemoglobin, and these the people who are in this situation where the red blood cells are full of only mutated form of hemoglobin. These people are the people with sickle cell anemia. Okay, so there are a lot of this protein in their red blood cell, in the erythrocyte, and they will tend to aggregate, and when the protein aggregate, it form a ball, like a bean. It continue to grow, and the red blood cells in the end start to look like a bean, like a sickle. So this is the way our DNA shape another time our lives. A single letter into the chunk of DNA, into our sequence of nucleotides, can make us sick and can make us heal, or I don't know, can make us a very good sprinter. So this is it for today, and let me know if this video was helpful, and see you later.